Brooke.
Yeah.
Um,
Oh, it started already?
yeah,
Just kidding. We play into that intro. What's up, class? Welcome back to another lecture on the senses. Today, we're going over our very last sense, which is vision. And yeah, we've had a good run, and it's actually our last lecture of the year, so it's very exciting. But before I begin, I want to introduce you to my special guest. We all know and love him. His name is Landon Cose. Give it up for my husband! Yeehaw! All right, let's get this thing rolling. So for our introductory question, I want to ask you, what is the most incredible thing you've visually seen? So just pause it, go around. It might be like a waterfall, it could be a puppy, it could be a piece of art or a statue. Please share the coolest thing that you've seen. All right, I'm out of breath from the wall ball. What is the coolest thing you've seen, baby?
I think I have this image of like my grandma's ranch and just like the sunrise over this pond and this like big she's a large field and just fishing out there.
Oh, that's really cool. I have like a visual kind of like that as my favorite one, and it was at my high school. All the freshmen went on this thing called the freshman river trip, and we went down this giant river that's in California for a week, and all of us went, and you weren't allowed to actually bathe or wash your hair or anything all week, which everyone was like, what? No way, we're not doing it. But you weren't allowed to bring soap to put into the river because it would pollute the river. So no one like bathed all week, and it was kind of hype. But we all went, and it was really fun. And there was this one night where there was like a thunderstorm. So we all hiked up to the top of this one mountain after we had docked all of our boats at night. And there was like the entire sky just like lit up with like lightning and thunder. And it was like really cool.
Oh, that's cool.
So that's probably my favorite. Okay, but we're talking about that because we're talking about the sense of vision, being able to see and perceive things. It's really cool that we're able to do that. It's very complex. So we're going to talk about that today. So we're going to start out with just the anatomy of the eyeball. So you guys all have a handout that has this eyeball. And the main thing that I want you to focus on top of even writing down all the notes right now, just make sure that it's labeled. And then you can go back and fill in whatever you missed later. So the first thing that we'll talk about is this thin, transparent, outermost mucous membrane that covers almost all of the sclera of the eye, so the white part of the eye, which we'll talk about. So that's this like kind of bluish, really thin, outermost layer. And the conjunctiva. Sorry, I'm having a hard time with words today. Conjunctiva doesn't actually cover this structure right here, which we'll talk about. That's important. So, have you ever heard of conjunctivitis?
Yes.
You have? What is it called? Typically, a layman's term for it.
It's just inflammation of the conjunctiva, so you have like a viral or like a bacterial.
Yes, and the lay person, unlike Landon, calls it pink eye. So pink eye, it's getting this outermost mucous membrane layer infected. So that could be from tons of things, like Landon said, problems in the body, pathogens, or not washing your hands after going to the bathroom. This will cause pink eye, and it's irritating and infecting that conjunctiva. And that's what's happening here. But we'll move on to kind of a layer deeper to that, and you see this white outermost layer of the eyeball. This is what we call the sclera, or the whites of the eye. So we have a picture pointing to that. What this does is it protects our eyeball and it maintains the shape of it. So that white part is the majority of the eyeball, and it's called the sclera. Now, when the sclera has a yellowish tint to it, like this poor guy has, we call it's jaundiced. That the sclera is jaundiced. Or this can also be applied to like our skin. Like if our skin has a yellowish tint, we could call it jaundiced. And do we know what this can be an identifier of? So if you have jaundice, What does that mean about something that's happening in your body? What do you think, Landon?
Thinking like a liver pathology or something that's like a hemolytic, something that's kind of attacking the red blood cells.
Totally. Something going on with our liver, maybe our gallbladder, or in our bloodstream. Can you explain, Landon, to them the mechanism of what's happening and why our sclera would get yellow from something like that? Can I turn it toward you?
Yeah.
And you can be brief with it. Doesn't mean
Okay.
like super long.
Your body has to get rid of red blood cells after they die after a few months, and so that when they die, they're kind of the heme in there is called unconjugated bilirubin. Ends up as that essentially. That doesn't get transported in the blood well. It's transported with albumin. Goes to the liver. Liver converts that to conjugated bilirubin and gets excreted. But you can have buildups of unconjugated bilirubin, which is has a yellow, essentially is yellow. And so the sclera in the eye has a lot of elastin, which that has an affinity towards. And so it's a really well perfused place, the eye. And so the unconjugated bilirubin will just attach to the sclera essentially and turn yellow.
Wow! Very impressive. Thank you very much, darling. So that's why liver or gallbladder not working properly would cause yellowing of the eyes, because that buildup of unconjugated bilirubin. Is that right?
Yes.
Cool.
And it also conjugated, but normally it'd be from unconjugated.
Okay.
Unless you have like a gallbladder thing.
Sweet. Thank you, darling. All right. So next up, we're talking about this outermost layer of our eye, which we know is the cornea. The cornea is transparent and avascular. What does avascular mean?
It doesn't have a good or no blood supply.
It doesn't have blood supply. A without vascular relating to blood. So there's no blood supply. And it's at the very front of our eye. Now this area is really highly innervated. So that's why when something scratches our eye, it hurts us so bad. It's very painful and it can be dangerous. But what the cornea does is it's the primary refractive surface of the body. So actually, Landon, will you just hit pause on the video? I want to grab something. Okay, I just grabbed a beaker. So if we fill this beaker, wow, that's highly pressurized water. If we fill this beaker with water, I'm trying to show you guys what refractive surface does. And we put a pencil into it. When you put the pencil in, can you tell in the video, darling, that the pencil kind of shifts? Like when it enters the water, the image of the pencil doesn't align with the pencil itself? Yes. Or should I bring it closer to the camera?
I zoomed in.
You can see?
Yep.
Okay, so what's happening here is when waves of light pass through a different medium. So right now they're in the air, but when they pass through a different medium, it actually slows them down, which changes their direction. So that's what's happening here. This is why the pencil doesn't look aligned with itself. It's because the waves of light itself and the image that it's casting, the direction of the waves changed when it went through the water. So that's why this doesn't align. Our eye actually does the same thing. The cornea, when light waves enter the cornea, it'll change the angle of the angle of refraction. It'll change the direction of the light waves. And those light waves will then end up somewhere on the back of our eyeball on the retina, which we'll talk about that. But right behind this cornea is a space that we call the anterior chamber. So that's this space right here. All of this. This space, which is in between the cornea and the iris, which we'll talk about, is filled with something called aqueous humor. It's a fluid-like substance. And this aqueous humor is there to maintain eye pressure, to keep your eyeball pressed out a proper shape, like this isn't gonna cave back in, as well as providing nutrition to the cornea itself, because as we said, it's avascular, so it doesn't have blood supply for those nutrients. So the aqueous humor itself provides that. Now, just like we have an anterior chamber, and I'm actually going to keep this up, we also have a posterior chamber. So anterior in front of, posterior behind. The posterior chamber is also filled with the same thing, this aqueous humor, but it's the space between the iris and the lens. So that's this space right here. So you guys can label both of those in your diagram, and just keep in mind both of them are filled with the same substance. Okay, now we can erase that. All right. Now, coming deeper into the eyeball, we have something called the iris. And we all know this because it's the colored muscular part of our eye. And what color iris do you have, Landon?
Hazel.
Hazel, yeah, I would say, what's the word I usually use for your eye color?
Brownish green.
Brownish green. Yeah, it's not like totally brown. You have like some yellow and green in it. And then my eyes are blue. So everyone has a different iris color that you could share if you want in class. But I looked this up. What's actually causing the color of the iris is the melanin. So you have eumelanin just like in your skin that causes it to be lighter or darker color, the brownish pigment. And then you also have phospomelanin, which is like the reddish yellowish pigment color. And interestingly enough, like your hazel eyes would be a mix of the eumelanin and phospomelanin together. Brown eyes would just be the eumelanin. But blue eyes are like almost a lack of melanin in that space. So similar to how you look up into the sky and the sky looks blue colored because it's just refraction and reflection of light off of different particles in the air. That's what's causing my eyes to look blue. So it's not actually a blue pigmentation, which I thought was interesting. But the iris controls the size of the pupil. And it does that through two different muscles. The circular sphincter muscle, which is the inner muscle in the iris, and the radial dilator muscle. So I'll show you those in depth. So you don't need to write those down. I'll show you in a second. So here we have the iris, which is the blue part. So I guess this would be like my eye. And then the pupil, which is just a hole in the iris, is the black. So over here, we start with the circular sphincter muscles. These are on the inside. And then on the outside are called radial dilator muscles. So when the circular sphincter muscles contract, they actually get bigger or thicker, which decreases the diameter of the pupil. So when these contract, they get bigger and it makes the pupil smaller. Now, when the radial dilator muscles contract, they do the opposite. They thin out, and so this causes even the inner layer of muscles to go out, which makes the pupil larger, dilates the pupil. So my question for you is, which muscle is controlled by the sympathetic nervous system and which is controlled by the parasympathetic? What do you think?
The radial is sympathetic and then the circular is parasympathetic.
The radial is sympathetic and the circular is parasympathetic. We already knew this because remember when we were talking about the autonomic nervous system, so sympathetic and parasympathetic, a sympathetic response would be fight or flight, like a bear coming at you and attacking you. And we know that this dilates our eye because we're trying to take in as much information as we can for the threat. So because the radial dilator muscles dilate the eyeball, we know those are controlled by the sympathetic nervous system. And then the inverse is true for the circular. Parasympathetic is rest and digest. We don't need all the information coming into our eyes, so our pupils can constrict and take in less information. Next, just the pupil. We already talked about this, but that's this structure here, and it's just the opening center of the iris. This is what actually allows light to enter into our eyeball. All right, next up, we have these little features that are called the ciliary bodies. Ciliary bodies, we have it on the top and the bottom here in this diagram. The ciliary body has two main components. First is the ciliary muscle. And these ciliary muscles actually will control the shape and the width of the lens itself. And we'll talk about that in great detail. What this allows for is something called accommodation of the lens. And we'll talk about what that means. But essentially accommodation is the lens changing its shape so that it can focus the image on the retina of the eye, which we'll talk about. But another part of the ciliary body is the ciliary processes, and these are the things that actually produce the aqueous humor. Where did we find the aqueous humor again?
It's in the anterior chamber.
Anterior and posterior chamber are both filled with aqueous humor, exactly. So, and it possesses the ligaments coming off of it. So that's what the ciliary body does. The main thing I want you to know is that it allows the shape of the lens to change. Now, I bring this up because in physics, whenever you take physics, if it's either here with Mr. Beaver or in college, you will learn a lot about the physics of light rays going through a lens. And it's really fun and actually like one of my favorite units in physics. But something you learn there is each of these lines is representing a wave of light, and this blue part is representing a lens, just like the one we have in our eye. When a lens gets thicker or wider, it will actually change where the focal point of an image is. Now, what does that mean? So, when the light rays come in through a lens, you will then get an image over here on this side of the lens projected from all those light rays. The focal point is when that image comes into focus. So, where the image is blurry of the, what is this, a parrot?
Yeah.
The parrot.
I'm
When the
pretty
image
sure.
is blurry of the parrot. I don't know why that just sounded like the wrong name. But where it's blurry, that'd be like all of this area. But when it comes into focus, that means that we're exactly at the focal point. And so this is what the lens of our eye is going to do. And it always wants this focal point to end or complete on the retina, because then that's how we get a clear image. If we can't focus the focal point on our retina, then our vision will be blurry, and this can cause vision problems. So let's talk about the lens. The lens has a biconvex structure. So we know that convex means like to go out, right? Concave means to go in, like a cave. So biconvex means it goes out on both sides. So that's the shape of our lens. And it actually allows for the best creation of an image. So it's advantageous that we have a biconvex lens in our eye. And it does this process of accommodation like I was talking about. So it's going to focus all of the light rays coming from wherever we want to focus on. So there's like a flag up there. Say if I want to focus on that, it's going to take all the light waves that are reflecting off of the flag. It's going to come through the eye, and they all want to concentrate on this yellow part of the eye in the back called the retina. Now, I wanted to throw in a couple of interesting things. Firstly, when an image gets projected through a lens, like our eyeball, it actually, through the rules and laws of physics and light waves, it will actually flip the image and invert it left to right. So what's being received on our retina is an upside down and inverted image. So what's coming into your eyeball right now is actually, Landon, can you flip the whole camera?
Like that?
Yep. This is really what your eye is receiving.
I just kept it.
Oh no, it like somehow, wait, it didn't show me upside down.
No.
That's so funny. Huh, that's interesting that the camera knows how to do that.
Yeah.
Huh, maybe just like our brain. Okay, so Landon flipped the entire camera to try to get me upside down and the other way around. But the camera knew that Landon was doing that. And so it corrected it within the iPhone itself. That's exactly what your brain does. Your brain knows that the image is coming in like this. And so it auto-corrects it basically when your occipital lobe takes in the visual information, which is super interesting. So then the question is, does anyone have to use glasses in the class? You don't have to say you do, but does anybody use glasses? I know Landon does, but he's shaking his head no. Landon, do you know if you're nearsighted or farsighted?
I'm nearsighted.
You're nearsighted. Okay. So what's interesting is in people who are nearsighted or farsighted, people who need to use glasses, is that focal point, that image, is not being focused on the back of the retina. So we'll draw a picture of this. So we have all of the light rays coming in, converging into the eyeball, right? And our lens then will take all of those light rays and converge them to make a focal point. What's happening with people who need glasses is that focal point is either landing in front of the retina or will continue out and actually land behind the retina. So this is why people have the blurry eyesight, is because the focal point isn't landing directly on their retina. So in nearsightedness, which is called myopia, it's landing before the retina, and that's because the eye, actually, the shape of the eye itself has elongated. So the lens is working properly in most cases, but the eye itself has gotten longer, like within the head. And so it's causing the focal point to end shorter than where the retina is. Now, this happens either because of genetic factors or certain disease or illnesses, or like age. As you age, your eye will grow longer, so that causes this. Or this may just be a problem with the lens itself. Most of the time it's the shape of the eye, but it could be a problem with the movement of the lens being able to cast the focal point onto the retina. So that's what's happening in nearsightedness and farsightedness, is we're missing the mark of where we want our focal point to be. But, and there's a lot more to that, so if you want to dive into it, it's very interesting. But let's continue on. So now we're finally to the retina, the thing that we've been talking about the whole time. This is actually the neural layer. So this is the layer that converts the light image that's being cast by the lens into a neural signal that our brain can actually receive. So at the very back of the retina, which is just this entire lining of this nerve tissue and cells in yellow, is something called the macula. And the macula, if I shine the light into your pupil and I was trying to look into your eyeball, this is what I would see. And macula is like a dense part of the retinal tissue. And the macula is specialized for sharp, detailed vision. So it's super interesting. The macula for sharp, detailed vision. And that's because there's a higher concentration of something called photoreceptors in the macula. So throughout this entire retina, there are photoreceptors, which are just light detectors. They're detecting light. And there's two types. One is rods and one is cones. Do you guys know what rods and cones detect? What do rods detect mainly, and what do cones detect? What do you think, Landon?
Cones are color and rods are actually light. So
Yes.
I was gonna say that cones to me are colorful. So that's how I remembered it.
Totally, yes. Okay, so let's start with rods. The rods, the majority of them are actually found in the periphery of the eye. So they're not in this back central portion. They're on the outskirts of the eye, is where the majority of their density is. And the main thing rods do, like Landon said, is they take in light. So they actually give you vision in dim light because they're extremely light sensitive. So even when a room is dark, if there's even a little bit of light, the rods will pick up on them because they're sensitive to light. But they do not have any color detection, and they can actually be pretty blurry. They're not super detailed. So they're mainly just determining like gross shapes of things and space. So we kind of sometimes talk about rods as being like our night vision goggles in a way, because they're really sensitive to light. So it's kind of, and then up here is just an image of us zooming in on the retina, and you can see that there's lots of rod cells, like 1, 2, 3, 4, 5, 6, like way more rod cells than cone cells on the periphery of the eye. Now, we also get to our cones. Our cones are more centralized right in the back of the eye by that macula region that we talked about, which is for the fine detail. And this gives us color vision and sharp detail. That's what the cones give us. Like Landon said, I remember cones because cones, especially ice cream cones, feel very colorful to me. So that helps me remember it better. But our cones actually only detect 3 colors. They're not even the primary colors, which is crazy. They detect blue, green, and red color. And the way they do this, we call them S cones, M cones, and L cones. It kind of sounds like small, medium, and large, but that's not what it's for. It's actually for short, medium, and long. So what's happening here, let me erase my image over here, is, I don't know if you know this about the frequency of light, but the higher the frequency, or the closer that waves are together, do you know what color this is more often?
Blue.
Blue. It's more blue in color when you have a higher frequency. So when you also have a high frequency, you have a shorter wavelength because like the frequency of a wave is inverse to its wavelength. So it's a shorter wavelength. So the blue cones are called short wavelength cones. Then as we get a little bit broader with our frequency, this will be more of a green color. And then if we have long wavelength, this is more red. So this is what each of these cones are detecting and how they can identify different colors. Now what our brain does is something called additive color matching, where it's taking the different cones and it's adding them on top of each other, and that's how we get all sorts of colors, not just seeing a blue, green, red world, right? Okay, now the last thing I want to mention here is that the macula, that space that gives you that high detail, where the majority of us, doing face recognition or reading occurs, in the very center part of the macula is a very cone-rich area, and this is where we have our highest acuity detail that we can see from vision. So it's almost like an hyper HD color camera in that area. So finally, we receive the light onto our photoreceptors. Our photoreceptors turn them into a nerve impulse signal. They send them to the bipolar cells, which is just a transitionary cell, and then finally to a ganglion cell in the retina. Those ganglion cells will converge. All the ones will converge at the back of your eye, creating something called the optic nerve, which is cranial nerve 2. And so this optic nerve will then carry all that information to the brain to be processed. But where all of those neurons come together is a space we call the optic disc. Because where all those come together, we're actually not able to have photoreceptors in that area because all of it's converging. So this is actually where we get the blind spot of our eye, is where if we're looking straight ahead, we can find a spot where we can't see our finger in our spatial field, because what's happening is the light information from that spot is refracting off of our lens, and then it's hitting the optic disc where there's no photoreceptors, where the optic nerve is coming out. That's why our eyeballs have a blind spot. And you guys are going to actually be finding your blind spot today in the lab after this lecture. So finally, the last thing we're going to cover today is something called the optic chiasm. And we're going to cover it very simply. What I want you to know is the information from your left visual field is processed by the right hemisphere. And all the information from your right visual field is processed by your left hemisphere. What's happening here is, maybe we can draw this and make it easier. Actually, this is pretty complicated, so I might just actually leave it at that. What time are we at?
29.
Okay, yeah, we're getting a little bit long. But essentially, your left visual field is going to the right side of your brain. Your right visual field is going to the left. It's kind of interesting, though, that these two fields are converging or staying on the same side, depending on what eyeball it's coming from. Why do you think that we have it like this? Why not just have all the information from this eye go to a side of the brain, and then all the information from this eye go to a side of the brain? Why are we switching some of the information over and some of the information stays the same.
I mean, it's almost like you're doubling up because there's still some that'll go into the same side.
Yeah.
The majority do cross over, like
Yeah.
you said. But I don't past that, I don't know.
Okay, so what I learned was with the optic chiasm is because we're taking some information from the same side, some information from the opposite side, and vice versa on both sides, is this actually allows for better 3D depth perception. It allows for better coordination of movement, and it allows our vision to be binocular. Like by having two eyeballs, it almost integrates the information way better than if each eye separately went to different parts of the brain, and then within the brain we'd have to coordinate all of that. But by switching some information over, almost like trading like snacks at lunch, we're coordinating the entire system so that it's almost seamless. Like when you look in your visual field, you don't really feel like you have two separate sets of information, even though you do. You feel like it's one set of information. That's because of this optic chiasm. So, thank you guys so much for being a part of today's lecture. I can't wait for you guys to check out this lab coming up, and I hope you guys enjoy it. See ya.